Our next speaker is Andrew Kornfeld. He is a graduate of UCSC, and during his time in Santa Cruz, he founded an organization called the Brain, Mind, and Consciousness Society, a student organization whose mission is to explore human cognition, behavior, and experience through student-led and student-directed education. He hosted a lot of different events there through the BMC, and is currently the founder and CEO of Practitioner Plus, an LLC that serves healthcare professionals in the Bay Area in a consulting basis, helping them to promote their missions and their presence in the web and social media and beyond. He does intend to attend medical school, and he's in the process of studying for those MCATs, and I wish him all sorts of blessings with that process. And he's going to speak with us tonight about neuroplasticity and speak a bit to how neuropharmacologically psychedelics work in the brain, and perhaps maybe help to shape and change our brain as we go. So let's give Andrew a welcome.
That. So, like I said, I am so excited to be here to talk to you about this subject. I'm in the process of studying for a really, really hard test called the MCAT, which is how you apply to medical school in this country, and it's really, really nice to be here to talk about something that I'm really interested and really, really passionate about. We're going to be talking about neuroplasticity, which is kind of like a really super hot buzzword that's been around for a while now, and we're going to be, it's really the brain's ability to change. And, I think some of you, I thought some of you were going to be like a lot of students at CIS, but I don't know, I don't know how many of you are students at CIS right here. Okay, awesome. We got a few of you. Some of you are training to be therapists, and I think a good understanding in neuroscience and a good fundamental understanding in science is really important, and I'm really, really excited to present. So without further ado, let's get straight into it. So what are we going to be talking about today? We're going to go over, like I said, some basic neuroscience concepts. We're going to be looking at, basically some common psychoactives and how they're working in the brain. We're going to be looking at a brief look at plasticity, especially on the kind of neuron level, because this is where a lot of the research is at this point. And we're going to be looking how this plasticity relates to psychedelics and entheogens. Okay. We're also going to be looking at, how this relates to our genes, and indeed how this, the larger conclusions that we can take for this. So, let's define neuroplasticity for one sec. It's really the foundation of what makes us human. It's how we learn. It's how we recast information. It's happening right now in your brain. Your brain is undergoing change, and simply neuroplasticity is the brain's ability to exhibit lasting change throughout life. So let's look at this awesome fundamental unit of the nervous system called a neuron. And let's just imagine a neuron like a tree. We have these branches right here called dendrites. We have an axon terminal, which we can think of the trunk of the tree, and we have we have this axon right here. We have the axon terminals, so we can just think of the axon terminals as the roots of the tree. And indeed, neurons work on electricity, and they communicate with other neurons, and electrical signals in your brain right now are transmitting from neuron to neuron. So if we, that's not the next slide. But before we get into that, I just want to bring up a quote from someone special to me named Gregory Bateson. Gregory Bateson was a professor at UC Santa Cruz. He was a cyberneticist, an anthropologist, a biologist. He was married to Margaret Mead, a very, very famous anthropologist. And he said in a very long manner, "You have probably been taught that you have 5 fingers. That is, on the whole, incorrect. It is the way language subdivides things into things. Probably the biological truth is that in the growth of this thing, in your embryology, which you scarcely remember, what was important was not 5, but 4 relations between the pairs of fingers." And he's saying, think not of the 5 fingers of your hand, but indeed think of the 4 relationships between them. So this can be extrapolated to neuroscience because it is indeed not the neurons themselves that we're concerned with. It is the relationship between these neurons that should be illustrated. So let's, for a second, take a neuron right here to explain what's going on. Let's say that this neuron is connected to a second neuron and a third neuron, and as you can see, they line up head to toe, head to toe. They're lining up from branches to roots right here, another one and an additional one. And all of a sudden, we've created a simple neural network. And what can happen is that one neuron can actually fire, or it can produce what's called an action potential. So we can illustrate that here. And let's say that this second neuron has a strong synaptic connection, okay, with this first neuron. This second neuron can also fire, and perhaps this signal can be propagated down through these neurons right here. Now, like I said, the connection is the key here, because not all neurons have this, extraordinary connection between them. Maybe this neuron right here does not have a strong synaptic connection and is actually weak, and so this neuron will not fire, and also maybe this neuron will not fire. So, really what's going on here, I don't want to go we're not going to go too deep into this, but we're going to be looking at some diagrams. So getting us really all on the same page is going to be really, really good. And explaining science is one of my favorite things. So what we're seeing an influx of positive charge in the neuron. So the neuron is really, really negative on the inside, and we have sodium, just like table salt. Table salt is Na+, Cl-, okay? And we have this cation, Na+, that's just flowing into the cell. And this is what we call an action potential. So let's just talk for a minute about complexity of the human brain. We have 80 to 100 billion neurons in the human brain, okay? We got 10,000 synaptic connections per neuron. So that makes a total of 100 trillion synapses. This is more synapses than are in the Milky Way galaxy. This thing is an entirely complex organ and has tremendous ability to change. I'll tell you, one other statistic. If you have 100 billion neurons in your brain, you have 10,000 connections per neuron. A big fan. Someone who I'm a big fan of is V.S. Ramachandran. He's a neuroscientist at UC San Diego. He says that if you have 10,000 connections per neuron, 100 billion neurons, you can take 10,000 and you can put that to the 100 billionth power, and that number, just based off that one element of the human brain, it can give us the number of possible brain states that exist, and that number is more than the number of known elementary particles in the known universe. So that's just one area of how much potential that this organ has to change. So I thought really quick I would just put a real picture of a neuron right here. This is one of the more, if you type in neuron on Google, you get this, and I will tell you that this is indeed a real picture. And how do we visualize this? What's going on? What's going on here? So scientists were really smart. We can take this protein that we found in jellyfish, and we can actually get the genes for this protein, and we can insert these genes into neurons in order to visualize them. And this protein is then expressed by the organism we are studying in some way, and we can see that light is reflecting off this special structure that forms in the middle of this protein called a conjugated chromophore. And that's how we visualize neurons. And I just thought, sometimes I get a little bit distracted, but I just thought for fun, I would also show you that, this isn't just the only color we can express. In the picture you saw in the beginning of my presentation, we saw all these different colors of cells, and, there's red fluorescing protein, there's cyan fluorescing protein, there's many, many, many colors that we can see. And this scientist right here actually got a bunch of E. coli and plated them on a dish and actually expressed a beach scene going. So, yeah. So back to the hard stuff. Okay. So let's let's like take a slightly zoomed in look at a synapse, like what's going on. The synapse is the space between, is how neurons communicate with each other. And I might just say that, neurons, they really, really like talking to each other, but they're not really the touchy-feely type. Of your 80 to 100 billion neurons in the human brain, not a single one of them is actually physically touching another neuron. They're actually only talking to one another. And so we've all seen this photo, or sort of an image like this before, probably, especially like on TV in the early 2000s. They'd often have ads for certain antidepressants like Prozac, and they'd be like, your depression might be caused by an imbalance of serotonin. Well, truth of the matter is we don't really know what's causing depression right now. But what's happening is we have this upper space right here, which is the axon terminal, which is like, let's just remember that it's the roots of that tree. The roots of that tree are squirting neurotransmitter out. Okay, so that's the presynaptic terminal, and that's squirting neurotransmitter out to the postsynaptic terminal, which is then eliciting some response in that neuron. And just to further illustrate this complexity, each one of these vesicles right here that we can see has thousands and thousands of neurotransmitter, whether it's dopamine or serotonin or glutamate or one of these neurotransmitters that we think of. And this is what one of those vesicles actually looks like. Just some of the beauty of biology. There is actually hundreds of proteins on the surface of just one of those vesicles. And this is from one of my cell bio books. And this is an artist's rendition of this vesicle. They did not even have the room to put all the proteins that are physically on this vesicle, whether they're involved with signaling or docking so, the human brain is immensely, immensely complicated. So let's talk about what, some of these neurotransmitters and get into kind of the meat of the talk here about how psychedelics and psychoactive drugs are working in the human brain. So oftentimes serotonin and dopamine, looks like dopamine's not even on here, get all the sort of they're like the super pop neurotransmitters. They're like the ones that everyone talks about. But in fact, the most important neurotransmitter is called glutamate. Glutamate is an excitatory neurotransmitter. It's involved in pretty much every single neuron in the brain. It's definitely involved in experiences with psychedelics or experiences with other psychoactive drugs. But what I want to say is that these psychoactive drugs harness the mechanisms of these neurotransmitters in the brain, and that is how we believe as scientists they work in the human brain. So, I want to go over generally how just give you guys an overview of like how psychoactive drugs are working in the human brain. And, we can think, in my mind, I like to think of it as two ways psychoactive drugs are working. So we can either pretend to be a neurotransmitter, and that's why I have a picture of a mask right here, because we're pretending to be something, or we can alter the concentration of neurotransmitters that are already in our brain. And the word endogenous just means sort of for yourself within, it's your own neurotransmitters or your own biology. Okay, so here's a drug that we talked about, that Natalie talked about briefly in her talk. We have cannabis. What's going on with cannabis here? Well, there's actually a neurotransmitter called anandamide, which is involved in many processes in the human brain, especially things like appetite. Big surprise here. Well, THC, tetrahydrocannabinol, is just hijacking this neurotransmitter and just pretending to be anandamide. So here we are sitting in this first category. Here's probably a drug that every single one of us took today, or maybe, at least 90% of us, caffeine. So caffeine is working in a slightly different way. We have an endogenous neurotransmitter called adenosine. Caffeine is blocking the receptor for adenosine, and adenosine, what it does is it makes us really, really, really tired. And so if adenosine can't bind, then we are stimulated. This is amphetamine, or Adderall. Adderall is working pretty much, on a few neurotransmitters like norepinephrine and dopamine, but is simply just flooding the synapse with dopamine. It's not having too much activity itself on dopamine receptors, so it's fitting into the second category of altering the concentration of neurotransmitters. This is cocaine. Cocaine is altering the concentration of neurotransmitters by blocking the reuptake. That's similar to also how antidepressants work, like a selective serotonin reuptake inhibitor. We're stopping the reuptake of serotonin. So here we have psilocybin. Psilocybin, as some of you might recall, is the active component in magic mushrooms. Psilocybin is broken down in the body to psilocin. A phosphate group is removed from this molecule. And what's happening with psilocybin is that we're really also in blocking the reuptake of dopamine and serotonin, but in a variety of unique ways. And I have it in the middle because it's also pretending to be serotonin and binding to a serotonin receptor. So MDMA, which Natalie talked about, is working in a lot of ways too. But again, we're flooding the brain with a lot of serotonin, a lot of dopamine. We're blocking the reuptake of these neurotransmitters. MDMA also has activity on the serotonin receptors and on the serotonin systems, and is activating a lot of other things. Like Natalie recalled that it's involved with oxytocin. Oxytocin is a peptide, which means that it's a little protein inside of us. And if you read my MCAT books, it tells you that oxytocin is involved with women's birth contractions, but they fail to tell you that it's actually one of the more important roles. One of the equally important roles of oxytocin is love and bonding, and is released in mothers when moms hold their babies, and when lovers kiss, and all sorts of things like that, and apparently during MDMA psychotherapy. So I put LSD in this first category here. LSD is binding to so many different types of receptors. It's hard to say exactly what's going on, but the primary receptor that's binding to is serotonin receptors. And you might think, we have this one receptor that we think about called a serotonin 5-HT2A receptor. But in fact, a really famous scientist named David Nichols, who is the chair of pharmacology at Purdue, or the former chair. I was watching a talk from him earlier today, and he basically is explaining how LSD is binding to a variety of these receptors, over like 10, 15 types of these serotonin receptors, and working on other neurotransmitters as well. It's really not very well understood how LSD is working, but I briefly put these two slides here from this awesome little website that I found that was produced by the University of Utah called Mouse Party. I would suggest all of you type this into Google. If you go to Mouse Party, you can find out how these psychoactive drugs are working in the brain. This is their explanation of LSD on a simple level, that LSD is binding to excitatory inhibitory neurons, which a lot of these synapses are different serotonergic synapses. Okay. So this is a diagram of a serotonin receptor. It's a serotonin 5-HT2A receptor, and this is the membrane of a cell. And we can see that serotonin is binding here, and this is what's called a G protein couple receptor. And what's happening is that these G proteins right down there, can I use my mouse? Oh, I can use my mouse. This is awesome. These G proteins are eliciting a certain response within the neuron when something like serotonin or LSD is binding to this receptor. So I had to ask myself, I was in college, I was studying neuroscience, and I really thought about the work that the Hefter Research Institute is doing and that MAPS is doing. And I was wondering that how it's possible that someone has an experience with a psychedelic like LSD or MDMA, and a certain amount of time goes by, okay? And then we see a significant change in behavior. We see a significant change in the psyche. Now, this can be an exceptionally positive change, or can be something negative, and some of you might disagree, but there is research that suggests that people who have an exceptionally negative experience with LSD in the wrong setting can develop things like schizophrenia, or different types of pathology in the human brain. But what I'm here to talk to you about, and what the research is suggesting, is that there are profound life-changing experiences. The research at Johns Hopkins with psilocybin and at UCLA is working with people just producing, exceptional mystical experiences. People claiming that this is one of the top 10, top 3 most impactful life experiences that they've ever had. We've seen that psychedelics, can be used in end-of-life therapy, that people are coming to terms with the fact that they have a terminal illness, that they are not going to be living on this earth anymore, and they've claimed that, I'm able to come to terms with this fact. We see in post-traumatic stress disorder, there's exceptional brain changes that are happening in the brains of people with post-traumatic stress disorder. We see reductions in a certain part of the brain called the hippocampus, which is involved with the conversion of short-term memory to long-term memory. And there's been exceptional research that shows that there's less synaptic connections and that neurons actually die in people who have PTSD when you put them in an MRI scanner. So their brain is undergoing some sort of plasticity. So how could it be that only 1 or 2 or 3 sessions of MDMA psychotherapy are eliciting this type of healing response? Well, I don't know. But probably something is happening in the brain. And I want to make this analogy that psychedelics and entheogens are not necessarily the same as a typical psychoactive drug. When you go see a psychiatrist in that manner, perhaps that psychiatrist would prescribe you some anti-anxiety medication like a benzodiazepine or an SSRI. That medication is needed every single day to elicit the type of response that they're looking for. And we found that they're not all that effective a lot of the time. A lot of the time, these SSRIs perform maybe only 3 or 4% better than a placebo does in their trials, and yet the FDA still approves them. Okay, and the thing is that when this psychoactive medication is stopped, usually, hopefully, a behavior similar, or a psyche, or the type of attitudes and mental capabilities, this person's mind is returning to a similar state that it was in before the medication was prescribed. What's going on with psychedelics? Something significant is happening here, and I really, really wanted to find out what it was. So I did a lot of research. And what I found, the first time I actually gave this presentation, I got up here, and that's where I ended it. I was like, something is happening, okay? And it was before I had taken a lot of my classes. It's before I had really understood what was going on in the brain. And I actually found out that there's enormous amounts of research. Well, not enormous, but there is substantial amounts of research that's showing that these compounds change the brain, okay? And I might add that a lot of them are not all in positive context. A lot of them are in negative context. But the authors of the papers don't necessarily consider the positive context that the change of the brain might be, what that might mean. So let's take a look at, what is neuroplasticity? Really quick, what is happening at a synaptic level? So one type of neuroplasticity is called long-term potentiation, okay? And what long-term potentiation is the strengthening of a synapse. And this diagram is like really scary here. But all I want you guys to take from this right now is that we are inserting more sodium channels in the postsynaptic membrane. So like we said before, those neurons can fire, okay? And the probability of them firing is based off how positive the internal environment of the neuron is, okay? So if we have more doorways for the sodium to come in, this positive charge, then obviously more is going to come in. Let's imagine that all of you were outside the room right now, and we were like, oh man, we really want to get into this talk because Andrew's talk's going to be really, really awesome here. So we just all got to get in. Only through that door, if all 80 of us in this room or so tried to flood in, it'd be really, really difficult. However, if we put a few more doors in, it would be a lot easier for us to get in. So let's just think this is all that's happening in this process of this synapse, is that more sodium channels are being inserted. There's a higher probability that this neuron is going to fire like that network that I showed you in the very beginning. How's everyone doing? Is everyone following me? Yeah? Got thumbs up? All right. Okay, so some of these factors right here, some of the stuff that we see is called nerve growth factor, or brain-derived neurotrophic factor. And what these are doing in the brain is they're actually helping sprouting the little twigs of the neuron. So we can see that when these proteins are around, they're really pretty, by the way. When they're around, we can see that there's actual growth in the little dendrites of the neurons. And they're called dendritic spines. So it's kind of hard to see on this exact picture, but there's a change in the microscopic neural anatomy of this neuron. So here's my evidence. If you don't believe me, LSD, DMT, these psychedelics are literally changing your synapses. What we can see here is that there's an increase in glutamate release that's happening from this neuron when LSD is binding. This is resulting in the overproduction of nerve growth factor and BDNF. Also, we can see that LSD, DMT, these type of psychedelics are directly binding to this serotonergic neuron right here, and increased BDNF is happening. I'm sorry, I misspoke. Just this cortical layer V neuron, but that little detail doesn't matter. What we do know is that when you have a plate of hippocampal neurons and you put some LSD on there, you put some psilocin or another psychedelic, like some sort of agonist of the serotonin 5-HT2A subtype family. There's a special one called DOI. We see rapid remodeling that's going on. We see these neurons changing before our eyes. So this is some crazy stuff that's going on. And I want to make another point about this before I make some concluding remarks. Again, I know I've kind of hammered in the basic science concepts to you guys. It's one of my favorite things to do. I really, really love talking about science, and I love teaching science. So you might recall from your high school biology course that you took 5 years ago, or 10 years ago, or 30 years ago, that the central dogma of biology is that DNA goes to RNA, goes to protein. What this means is that our genes are being read, and this is translating into who we are. And I just wanted to give just a quick analogy of, this is the class I taught at UC Santa Cruz, and how I explain it to my students that I taught was that, let's imagine that we're at a giant dinner party, and the DNA is the giant cookbook that we have for all the different recipes that we're going to be making. Each piece of RNA is a single recipe that we have. And we know that a single recipe only applies to a single dish that we're going to be making. And in fact, each piece of mRNA, it is called, applies to each protein. This is a special protein I put up there. It's called myeloamine oxidase, or MAO. It's involved with the breakdown of neurotransmitter. It's indeed one of the components of ayahuasca and how DMT is active in the brain. MAO is an enzyme that breaks down neurotransmitters, and MAOI is something that inhibits that enzyme. And one component of ayahuasca inhibits that enzyme and allows the DMT to be orally active. And many antidepressants work this way. It's not a good thing to ever mix with psychedelics, but it's deeply involved. So the point that I'm getting to is that what we learned in high school about how DNA goes to RNA goes to protein is kind of only partially correct, because we indeed can turn on and off our genes. Every second inside of your brain, there are genes that are being turned on and off. And if you don't believe me, you can take this guy's word for it, which was, I would highly recommend all of you read this book. It's called The Brain That Changed Itself. It's a New York Times bestseller. Scientists used to believe that the brain was sort of a more static entity, that even as early as the 90s, okay, that your behavioral patterns, that your pathologies were stuck within inside of you. We now know that's just so not true at all. And while Norman Doidge in The Brain That Changes Itself does not talk explicitly about psychedelics, he illustrates this point about genetics. He says most people assume that our genes shape us, our behavior, our brain anatomy. He's referring to a scientist named Eric Kandel, who is a psychoanalyst and psychiatrist, who was one of the first people to discover plasticity in the brain. He said, this work shows us that when we learn, our minds also affect which genes in our neurons are transcribed. Thus, we can shape our genes, which in turn shape our brain's microscopic anatomy. And if you don't believe me, take my neuroscience book's word for it. And we can see right here, don't worry, really, really scary diagram. All you got to know is that that's the nucleus of the neuron that's going on. There is transcriptional regulation. Genes are being turned on and off. And yes, synapse growth proteins, that BDNF and that NFG we were talking about, that should be NGF. So yes, I'm telling you that LSD and DMT and psilocybin is turning on and off your genes inside of your brain and is changing your brain. Okay, so that was kind of a lot of information that I spat at you, and I deeply wish that I had more than 30 minutes for this talk. I could probably teach an entire class on this subject because there's so much to learn about it. And I feel that in some ways I gave you a gross oversimplification of how the brain's working and how this is working and how different drugs are working. But I want to make a couple points about this. So every now and then in Western medicine, we have certain procedures or medications that can actually cure illness. We don't think about this that much, but if you have certain infections, you can take antibiotics and the microorganisms within your body will die and you will heal from that infections. Yes, antibiotics makes certain things worse, but the point holds. If you have something like appendicitis, you can go have a surgeon remove your appendix. Now, what do these things have in common? Okay, if we consider the body and the microbes within the body, or the body with the appendix inside of it as a single unit, there's been an event, whether it's been the surgery or the administration of antibiotics, and then there has been a change within the body. And we have seen the healing take place, and we have seen curing. How is it possible that someone with PTSD receives only 2 sessions of psychotherapy with MDMA, and then all of a sudden they no longer exhibit symptoms of PTSD? I'm trying to make the analogy that this might be a type of somatic plasticity, a type of plasticity of the body, but this does happen in nature, and psychedelics may very well be doing this. And I was studying with, I don't know, do any of you watch Khan Academy? Ever learn on Khan Academy? Okay, we got one. Awesome. You guys should totally learn on Khan Academy. It's really great. I was studying for my MCAT, and they were talking about genes getting turned on and off. And they were talking about how outdated this sort of phrase is about. I wish I could claim responsibility for this myself, because this is so cool. But how outdated nature versus nurture is. Because what we're learning is that it's not nature versus nurture, it's indeed nature through nurture. Indeed, that the experiences that we're having, whether they're in our everyday lives or whether they're with psychedelics or under the conditions of psychotherapy, are indeed turning on and off our genes and thus shaping our behavior. And I want to end on this note. There are scientists right now who are tirelessly working to map every connection in the human brain. This is their goal. There's 100 trillion synapses in the human brain, and this differs from our genomes, okay? You know, all of us share 99.9% of our DNA. So they're pretty similar. So the amount that our DNA differs from one another is not all that much. The way that our neurons are connected and the internal environment and the complexity is vastly different. It is like an impossible task, okay? What they've managed to do is they've managed to map the connectome of a simple slug, okay? And even doing that was like, it took like years for them to do that. So what I'm trying to say is that if we could like map every connection in the human brain, and then we could identify exactly what that synapse is doing and exactly how that synapse relates, like Gregory Bateson was saying, the connection or relationship between another synapse, then, you know, it's possible that we could find the location of PTSD in the brain. We could find the location of depression. We could find the location of, what's hurting us. We could find the location of what's stressing us out and leading to autoimmune conditions, which is a whole nother talk for you guys. But that might be thousands of years away. Why are we afraid? You know, we see that it's possible we have this tool right now that we're just a little bit skittish to use. So why should we be waiting thousands and thousands of years, which is going to be amazing when it happens, when we have this very, very powerful tool with us here right now? And I think that understanding that, you know, there's a lot of research going on with MAPS and Hefter, and understanding that these substances change the brain in the manner that they do gives us a tremendous amount of power. And it gives us a tremendous amount of responsibility to use these substances wisely. And I think it shows us the true potential of what psychedelics can do. And it's been a pleasure talking to you guys. So thank you so much. So I feel like I spat a lot of information at you guys. So please feel free to ask me anything you'd like.
Yeah. Let me bring the mic to you.
Okay, this may be too complex of a question, but I don't understand exactly what turning on and off genes does and what the potential that has for PTSD or changing behavior.
Sure. Well, let me do my best to answer that. So when you guys are listening to me right now, okay, your brain is undergoing change. The way that your brain undergoes change is by turning on and off genes. There's genes that are like, yo, make my neuron grow, okay? And that's what's happening. The reason why it's significant is because it's another case of, you know, us thinking that the genome is static and this sort of like, you are your genes. You are the product of your biology. Well, yes, you are the product of your biology, but guess what? Your biology is super changeable, and it's even more changeable under the context of a psychedelic-induced experience, especially when that is accompanied in the correct environment, like a therapeutic setting. Does that answer your question?
Yeah, that was so cool. Thank you.
Okay.
You mentioned that you were talking about it changing our brain, and you showed the slide with that in level 5 or something. And so there's a couple ways to interpret that. One is that, taking the substances would just kind of change all the neurons in that level. Or it could be that, yes, they are changing them, but that's dependent on the experience that the person is having and which neurons are getting changed and how. So which is the case, or do we not know?
Let me see if I understand your question. I think your question is about environment. Is that what it's about, like the context?
Well, one is it's kind of a chemical phenomena that's just happening. The other is that it's actually mediated by what's going on, such as setting and the experience, the direction, the intention that the person is having.
Sure. I mean, I think what Natalie alluded to this is that psychedelics are catalysts, are catalysts for processes, and it depends fully on the context by which we're in, and whether the biology is coming first inside of us, or whether the setting is coming first, setting is coming first. It's it's really hard to say, but I would hypothesize that if someone was given MDMA in a really, really scary environment, that it probably would do more harm than good. Did that?
Yeah, different things happening in the different neurons.
Yeah. Yeah.
My experience in this world, and being a neurophysiologist, I have a pretty detailed understanding of the neuropharmacology involved, and I read papers and I learn and all that. You got to have the equipment in place. You got to have the receptors. You got to have the internal messengers. The hardware has to be operational for these molecules to act.
Yeah.
I've done many ceremonies and I've met many people who just can't get high on whatever you give them, or they need an enormous dose to get high. And I ask myself, what is it that's different about these people? And in the perfect setting and everything.
Okay.
And I've come to the conclusion that they have a different neurochemistry. They must have. Or they have very strong mechanisms of, psychological blockages that really just prevent them from letting go.
Yeah.
So,
do you have a question?
no, it's just, this is a very interesting conversation. I mean,
It is.
And I work with DMT quite a lot, and I know quite a bit about it.
Okay. Well, thank you so much for your comment. I think just one comment that I have on that is that, yeah, everyone is different. And we know everyone's different. Maybe it's because each of us has 100 billion neurons in our brain and they're all doing different things. Okay. Lots of other questions. Can we take any more? Or?
I have one comment to that, and that is that it's not just about neurophysiology and neuropharmacology. I mean, these substances are being metabolized through the liver, and depending on the expression of cytochrome P450 enzymes that are responsible for the metabolism of most drugs and psychoactive agents, that can greatly influence how someone metabolizes something. So if someone over or underexpresses a particular enzyme, they may metabolize the heck out of something, and it might clear out of their body more rapidly, or they might have a higher level of something in their system, and therefore be more sensitive to maybe a dose that the next person might not feel at all. So that's, there's a lot more to the picture.
Dude, Dr. Natalie to the rescue. One day I'm going to be as cool as her.
That's true.
I'm currently dealing with some glaucoma, and I just recently also just heard somebody speaking about neurogenesis, and I'm getting a very definite sense of the eyes and the brain as being very much of the same thing, and how one of the things I'm experiencing personally is having lost the light when I look around is very depressing. There's it's very connected emotionally. So I just was wondering if you wanted to comment about eyes, because I see EMDR showed up and a couple of eye things. Do you have any comments about eyes?
I don't have a huge amount about eyes, but I do have one thing about neurogenesis to tell you.
Okay.
Neurogenesis means the production of new neurons. And it was once thought that did not happen at all in our brain. And I would say that the primary healing of psychedelics and stuff like that is probably not primarily through neurogenesis, but in fact the rearrangement of neurons. And it's been shown that people have lost like half their brain. little children who have extreme seizures. And unfortunately, the doctors did not know about CBD or were not accepting of CBD for their seizures, but they actually would take out an entire hemisphere of the brain. These kids actually live like pretty normal lives, and they go up and they're great. And when you look, they have half their brain. How is that possible? Well, it's not about the number of neurons. It's not necessarily about the neurogenesis, but it's about the way they're connected. I'm sorry I don't have more on the eyes, but I could think about it and get back to you.
Is the nerve growth factor you mentioned the same one that's in mouse saliva and snake venom? That
No.
there was an Italian woman scientist that discovered. It was
I'm not
called
sure.
Nerve growth factor.
If it's called nerve growth factor, then it's the protein that promotes growth of
'Cause-
neurons. It doesn't promote to my understanding, it doesn't promote the replication of neurons, but it enhances
Promotes
their
the
growth.
Yeah.
Yeah.
So I had a pet rat who used to lick me all the time, and if you could get him to lick something, it would get well twice as fast or 10 times as fast. It was very, very healing.
Got some nerve growth factor
I don't
going.
know if I can get my eyes licked.
Let's take one more question, and then we're going to take a break.
Our bodies have a tendency towards homeostasis. Do our brains have that same mechanism?
You said, sorry, I just missed your question. You said our bodies have? What did you
Have
say?
a tendency towards homeostasis, and our brains have a general neurophysiology that's going on. Is there some type of norm that it sets to, or do
Physiologically, yes. In terms of the connectivity, no. There's no norm.
All right.
We had one
All right,
one.
we'll
super
take one more,
eager-
and then we're
eager
going to
guy
take a
on
break,
the-
and we'll have a panel at the end, so you'll have time to ask more questions with everyone.
So this is something that I thought I heard intimated, but I'm wondering if you can just confirm or deny. Are you suggesting that the neurons that have the BDNF stimulated in them, or that where opening of doorways are encouraged, or are the ones that are active during the experience, or could they be? What you said about MDMA in a bad setting made me think of that. But I didn't know if you meant to say that outright, or if you're not. We don't actually know which ones are being encouraged.
Well,
to grow in
obviously,
that area.
this is from a Nature review paper by someone who I respect very much, named Franz Voldenweider, and he's a researcher with the Hefter Research Institute. And obviously, yes, all of these neurons are involved in a psychedelic-induced experience. And I'm not going to go into the brain parts on a larger scale level. I didn't have time in this presentation to go the larger brain sections that are being activated or turned off in a psychedelic experience, which that's like a whole nother discussion and is so interesting. And but the answer to your question is all of these neurons are being activated. Okay.
All 100 billion?
No, in this diagram, I mean, the notion that obviously that you're only using 10% of your brain, that's not true. You're using all sorts of percentages of your brain and all sorts of different percentages of your brain at any given time.
Only the neurons that have the receptors are the ones that get activated. The other neurons are not getting directly activated. They may get indirectly.
Sure. So it's Juan, right? It's Juan. Dr. Juan pointed out that that there's neurons that are being directly activated, like this 5HT neuron is getting directly activated right here, and then we also have this cortical layer V neuron, which is getting directly activated, but this deep cortical neuron right here is only getting indirectly activated. But a small little detail, as is all biology. It totally sucks. There's like so many little exceptions in biology, and you have to learn them all. But okay, thank you so much, everyone.
Thank you, Andrew.
Appreciate it.